Hi guys, today we're going to be talking about screens and selections. If you guys work in a bio lab or do anything related to bio, chances are you probably are going to be doing a screen or most likely a selection at some point in your career. Some more than others. I do a selection every day whenever I'm plating bacteria. So they're very similar techniques, but there's a few fundamental differences. So let's just talk about, let's start with a screen. So screen, oops, pen's not writing, screen. So if we start off with BFP, blue fluorescent protein, and we want to, instead of having a blue fluorescent protein, let's say that we want to get like a pink fluorescent protein, so PFP, which I don't even know if that exists. Probably not. What we would do is we'd make a library and transform it and plate it. And so here's our plate, and then we'll probably get a bunch of colonies. A lot will probably just be dead or blue or whatever. And we're going to be looking at them. Realistically, we're going to be looking at about 10,000, 100,000 colonies or plates or something like that until we finally find a pink fluorescent protein colony. And the act of kind of sifting through all of these different possible constructs and actually looking for the phenotype that you want is the screen. So the output of a screen is a phenotype. So screen means you're looking for the phenotype. And so in this context, in microbial biology context or genetic engineering context, that means you're looking for maybe a change in color, like a pink fluorescent protein, or if you're looking for an enzyme pathway, maybe you're looking at the yield, so maybe you have to run a GCMS on a bunch of different samples, and you're actually looking and inspecting every single colony. So it's painstaking, it sucks, you have a ton of colonies, you know, if you have 10 to the 9th individuals per mil, you do not want to look through every single one of those. It's actually impossible. But you know that's what we have. We have to use that, and it works, and we've used it to great effect. A selection is a little bit different. Selection, you're not looking at the whole population. So in the screen, you're looking at everything. You're plating everything. You're sifting through it, and you're finding the needle in the haystack. The selection is a little bit different. And I like selections a lot. They're real cool. The selection, the output of the selection is not the phenotype, but rather it's survival. So if I'm looking for, so this is not a really realistic example, but let's just say that we had a selection for pink fluorescent protein. Somehow the pink fluorescent protein, the presence of that pink fluorescence would allow a cell to survive, and the absence would mean it would die. So if we're starting off with our blue fluorescent protein, and we make our library, and we're looking for that pink fluorescent protein, and we transform this library, so remember we have we'll just say for argument's sake that we have 100 library members in this blue fluorescent protein library. Realistically, we would probably have 10 to the 10th or something like that. But just for the sake of argument, we'll just say we have 100 library members in this blue fluorescent protein library. We'll transform those in to our cells, and then we'll plate our cells on our selective conditions, and anything that we get that grows should be pink. So we'll just say we get 2 pink colonies. So what happened here is the only thing that could survive on our selection is what we want. So another way of saying that, the output of the selection is survival or cell viability. So this is kind of a silly example, but something that we do often, pretty much routinely, is we're selecting for antibiotic resistance. So let's say we have a plasmid, and we want to put some gene that we want to study inside of our bacteria. So here's our bacteria, here's our plasmid. So we can transform it, but we only want to see the bacteria that receive that part. So a very easy way to do that is, in addition to your part, you can just include some sort of antibiotic resistance. So we'll say this little thingy right here, that's our antibiotic resistance. So that if our little bacteria gets that plasmid with the part, it also gets the antibiotic resistance. Whereas if we have this guy's little neighbor, this brother, this is another bacteria, if this does not get the plasmid, it does not have that resistance. So then if we take both of these bacteria and we plate them on our selective conditions, so we'll say that we're plating them on an LB agar plate that has this antibiotic in it, then this antibiotic is going to kill the bacteria that does not receive that plasmid because it can't make that antibiotic resistance. Whereas the bacteria that does receive this plasmid does have that antibiotic marker, it will produce the antibiotic resistance and it will survive. So it is selected for. So the output of this antibiotic resistance selection is the survival of bacteria that take up that plasmid and can express that antibiotic resistance. So that's a selection in a nutshell, and they're cool because you don't need to look at the tens, hundreds, thousands, millions, billions, trillions, however many cells. You don't need to look at the entire population in order to find what you want. You're only looking at the ones that survive. So genetic selections are awesome. Unfortunately, it's hard to make a selection. So you can imagine it's pretty easy to make a selection for like antibiotic resistance. You just put some antibiotics in there, and if you have an antibiotic resistance marker, your thing survives. If it doesn't, it dies. But this silly example that I talked about with PFP, with our pink fluorescent protein, I can't think of a way that you could select for pink fluorescence. But it's very easy to find pink fluorescence physically.